Hey everyone, welcome back to another DNA Master tutorial. Today I'm going to be showing you guys how to prepare your final files for submission to the quality control after you've finished all of the steps of your annotation for your phage and you want to submit it. It's a pretty exciting step. You know, you've done everything. You've deleted genes, you've added genes, you've changed start sites, you've called functions, and you've made it through the entire genome. Now you have a better idea of how your phage is set up and how its genome is oriented, and I think it's a pretty awesome thing. So I'll show you guys how to properly submit your DNA master files once you're finished. So the first thing you're going to do when you're finished everything, when you added all the notes into every gene, the first thing you're going to do is go up here to the documentation tab. You're going to recreate the documentation, and this will take a second. After it's finished recreating, you're just going to save a copy of it. So we're going to go to save as, and we're just going to you should save it into the folder that you've been saving it into all along. I'm just going to say Colucci 29 copy, just in case something goes wrong and we just have a copy of the final file that we're going to use. So that was the point of that. Now we're going to go back to the features tab, and what we're going to do is validate our genes. So when we click this button validate down here, it's next to post. What it's going to do is check everything and make sure all the start and stop sites actually start on a start codon and stop on a stop codon, and make sure everything else is in order. And if you get any errors, it'll tell you what the errors will be and where the errors are located, so you can easily deal with them and fix those before advancing. But everything needs to be validated before submitting. So here we go, we're going to click it, and as you can see here, a little window popped up. It says all open reading frames appear valid, which is a good sign. That's what we want to see. Now the next thing you want to do is make sure that if you deleted or added any new genes, that these numbers here on the left, you'll see they'll be off a little bit. So for example, I deleted gene number 93, Colucci 93. Obviously you can see it is missing. It goes from Colucci 92 to Colucci 94, which means I got rid of that gene in between there. So I got rid of, I think, 5 total genes in this genome. So we definitely need to renumber them so that all of these, so that it will show up in order down here, so it won't skip over 93. So to renumber them, all you have to do is simply go to the bottom here where it says locus tag prefix. You're just going to type your phage name in here, and what you're going to do is just hit the reassign gene data, and then hit yes. And what that will do is just pretty much save all the data and just renumber these genes so that they're in the correct order. And you'll see in a second after it's done updating that we no longer have 117 genes, we have 112 because we deleted 5. So that makes sense. And now we have everything renumbered, which is good. Alright, now on to the next step. We can head back into the description here. The next step in preparing your final files is to make sure that if you merged files, so the merging feature was added so that work could be split up over a group of people. So for example, let's say a class wanted to do a whole genome and they were each assigned 5 genes or something like that. It would be easier to combine the DNA master files into one file and combine everyone's work into one final project. So if you use that feature, what the merge feature will do is if there are 2 sets of notes for the same gene, so say somebody messed up and they got their numbers wrong, and they annotated genes 1 through 5, but their teammate or their classmate forgot what their gene numbers were. They thought they were 1 through 5 too. So there was 2 people that ended up annotating genes 1 through 5, which means that when you merge the files, genes 1 through 5 will have 2 sets of notes. So it will appear like this, and then there will be some sort of divider, I forget what it is, and then it will have another set of notes below it. What you want to do is make sure that every single gene in your genome has only 1 set of notes. So if you use the merge feature, make sure you go back and check to see if every gene has only one set of notes. If it has 2, the annotation will be sent back to you upon submission the QC analyzer, I guess, will look at your file, and they will let you know that something's wrong and that you can't submit it. So if you utilize that feature, make sure that every gene has one set of notes. Otherwise, if you didn't use that feature, you don't have to worry about it. Alright, so there are two more things that we have to do before we can finalize this file. So the biggest thing that probably takes the most amount of time is you're going to have to reblast all of the genes in the genome. So you know, it may have taken you a week to complete this annotation, or it may have taken you 2 months, it may have taken you 6 months. But the point is, in between the time you started and originally blasted all of the genes in your genome, and between that time and when you finished the annotation, the information in the database, in the protein blast NCBI database, could have changed. It could have been updated, and it could have been, you know, some more information or publications could have been added that suggest gene features, or excuse me, gene functions for some of the genes in your genome. So regardless of how long it took you to annotate this genome, you have to reblast all of your genes. So if you go up to your first gene, you click on the first gene, you go to blast, the blast tab. What you can do is actually hit delete all, and by hitting this button, it's going to say delete all blast hits for this feature, and you're going to hit yes, and it's going to delete all the blast results just for this gene. And then this menu will pop up. It says there are no blast results for this data. We're going to hit clear all, which means it's going to clear all blast results for all genes, and we're going to hit yes. Now this will take a minute or two because it has to go through each gene and manually delete every entry, but just to be safe, we're going to do that. Alright, now that all of the genes are cleared, we're going to hit blast all genes, and this window is going to pop up. It may look familiar from when you from after you auto-annotated and you blasted your genes. Then we're gonna have to do it again, and like I said in the first tutorial showing you guys how to do this, it takes a long time. So I would suggest doing it during off-peak hours. You can look down here in the bottom left, it will say green off-peak hours, which means that, you know, it's the least amount of traffic during these hours, 9 p.m. to 5 a.m. As you can see, I'm doing this at 11 p.m. at night, which is good. We're in off-peak hours, so it may go a little bit faster, and you know, the database or the server may be able to take larger batches, so it should be good. So we're gonna hit blast all, and it's gonna commence the blast result, and then we'll have to wait till it's done. This may take a little bit of time, definitely. So if you are doing this on, you know, a school computer or something, you may have to leave it running overnight, or, you know, something like that. It's going to take at least a couple of hours depending on how many genes you have. So for the sake of this tutorial, I'm not going to reblast all of the genes. I did 2 days ago when I resubmitted. So if we go to the blast tab, it'll say the date, which was 2/22. It was 2 or 3 days ago. That was good for when I submitted it on the 22nd. But after everything is reblasted, what you're going to do is go through every single gene one more time and make sure that the top blast result here matches the blast result in the notes section. So we'll see here, for example, hypothetical protein in ARV1 matches 1:1, and we'll go to the description, and there we go. Hypothetical protein ARV1 1:1 match. So we're going to do that for each gene and make sure that none of the blast results were updated or changed, and that all of the functions are still correct. So that might take a little bit of time, maybe a half hour to an hour, but it's definitely worth doing before submitting, because your blast results may change, and it may change ultimately the function of the gene. Once you're done that, you're pretty much ready for this file to be done. So there's a few more things. Make sure you still have the original bioinformatic originally generated information up here. This first line, original glimmer call at base pair blah blah. You want to keep that in here for each gene, so it's going to be in there for each gene. You want to leave that in there for the QC process. Also if obviously if you had a function, you want to have the function listed here next to function, as well as in the function box. So let's see, we'll go here. This has a function tail sheath protein. It's here listed in the notes as well as the function box. And just make sure those are all squared away. And then once you're done, once you think you're ready and you've completed all those steps, you're going to hit post one more time. You're going to go up to documentation and you're going to recreate the documentation. This time we're going to name our file something very specific, which is used in their database to organize and just keep track of the files that they have on hand for DNA Master. So the document is recreated, documentation's recreated. You'll notice that the genes are renumbered, so that's definitely good. And we'll go up to File, Save As, and we're going to rename the file your phage name _ complete notes. So that's what your file is going to look like. So it's going to say your phage name _ complete notes, and then we're going to hit save, and that will be that file. That's your complete note file. That is what the file is. This is the file that's going to be uploaded to PhagesDB. So anyone, once you submit it, anyone will be able to download this file, look in your gene notes, and they'll be able to tell what you named the protein functions, and where you got that information from, etc. They'll be able to see all your complete notes and everything like that, which is awesome. So that's one of the files that you'll have to create upon submission. So there are 3 more things you have to do before submitting the genome annotation. This is just the complete notes file, but in the next video I'll show you how to create your minimalistic final file. So definitely save this one. You need this one to submit, but you also need 3 more things. So I'm gonna make these separate videos because this one is a little bit longer. But thank you for watching. I hope you learned something, and I'll catch you guys in the next one.